So, welcome back, those of you who have been with us for the past couple days, and also welcome those who are newly joining us. My name's Eric Decker. I'm in the Department of Food Science at the University of Massachusetts. I'm also a member of the Food Forum and the chair of the planning committee for this workshop. On behalf of the Food Forum of the National Academy of Science, Engineering, and Medicine, I would like to welcome you to our virtual workshop. If you're joining for the first time, the workshop is hosted by the Food Forum, which is a convening activity in that National Academy. For more information about the Food Forum, please visit our website. You can also find the agenda and the bio sketches of all the speakers on the website, as well as the membership of the Planning Committee. If you missed the last 2-day sessions, the workshop is being recorded, and presentations and videos will be posted on the website in about 1 to 2 weeks. We will continue with session 3. I have the pleasure of welcoming back yesterday's moderator, Bruce Lee, and Bruce is going to give us a summary of the previous 2 sessions. Thank you.
Thank you, Eric. Okay, well, hopefully everyone can see my screen right now. So I'm going to be summarizing largely day 2. Cindy gave us a great summary of day 1, so I'm going to dovetail upon that. And again, wanted to thank all the speakers and panelists and organizers of this workshop, including a big shout out to Cypress Links and Heather Cook for their guidance. Okay. So basically what we want to do essentially is, with this workshop, is point out what some of the specific issues in precision nutrition are, and look at this chasm that still exists between different types of nutrition issues and technology. And how do we bridge this chasm, and how do we help technology address a lot of precision nutrition issues? So that was largely day 2, and then day 3, as you'll see, we'll really be looking at some of the overall issues when it comes to implementation and translation of precision nutrition. So we really wanted to move towards the specifics. Some previous precision nutrition workshops have introduced the idea of precision nutrition, but day 1 really looked at a lot of the specific issues that are part of trying to achieve precision nutrition. Day 2 gave some specific examples of new data science, computer, and tech approaches. And as you'll see in day 3, we'll be looking at specific issues of implementation and translation. So we started off day 2 with a great overview from Marriott Abrahams from Kina. And she basically looked at what the current landscape is for tech when applied to nutrition. And she discussed the origins of the tech industry's approach to nutrition. Many of the early attempts had limited evidence. It was really a niche market with a few high-tech players. You had some early adopters, which were largely female, educated with disposable income, so very focused demographic. Behavior change was sort of a side note. But things have evolved, and actually the COVID-19 pandemic over the past year has really driven demand for telehealth, smart eating apps, etc. So in many ways, it's accelerated the movement towards precision nutrition. We're seeing new trends like microbiome, cardiometabolic health, hybrid approaches, and then questions about sustainability, and then regulation certainly is looming in the forefront. And we're seeing new entrants such as consumer health and retailers. Then we moved on to the different panels. And we organized the different panels according to scale in terms of the approaches that they were largely using. But we have to emphasize that precision nutrition means crossing different scales. This was just a way to organize the day. And Denise Ney from the University of Wisconsin-Madison gave us an example of using newborn screening for inborn errors of metabolism with whole exome sequencing. Jim Kaput from Vidiant showed how you can stratify people based on their polygenic risk score, and then determine how these different types of characteristics ranging from dietary intake to nutrient levels to social demographics can be distributed among these different strata. And Ahmed Al-Sohaimi of the University of Toronto and nutrigenomics showed that when you give people the right genetic information coupled with actionable items, like things they actually can do, he felt that it can actually motivate behavior change. And then we moved on to the biological physiological scale. Sarah Berry of King's College London showed how they've been doing work on creating a dashboard that can predict individual postprandial response to food, and then developing at-home tests to help determine what kind of personalized dietary advice people should get. Mikhail Ryan of Weizmann Institute of Science talked about evaluating the clinical impact of personalized postprandial targeting diets in populations with imbalanced glucose tolerance. And Guru Bhanavar of Viome talked about analyzing gut microbiome activity and categorizing foods, and then recommending specific foods to specific individuals based on their microbiome activity as well as other characteristics. Then moving on to the individual scale, Andres Acosta from the Mayo Clinic showed how he's used machine learning to identify 4 different obesity phenotypes. Samantha Kleinberg of Stevens Institute of Technology gave examples of how her team is using sensor technology to show what people are eating and when, such as based on their movements and based on different types of characteristics that you can help infer diets from. And Diana Thomas from the United States Military Academy at West Point showed how he can use natural language processing to help better understand what people are actually thinking. So many times, their initial words may not be so obvious, but you can really kind of put together these complex relationships. And then finally, we covered the social ecological scale with 3 great examples. Kayla De La Haye from the University of Southern California showed how using social network analysis can really better understand people's relationships and how they're connecting with each other and how that can influence diet. Sean Duffy from Omada Health showed how combining data collection devices and analytics can then be used to generate more tailored specific recommendations for obesity and weight control. And Michael Howe of Google Health gave examples of how you can conduct extensive mapping geospatial analysis to better understand neighborhoods and their characteristics. And of course, these are being done in other disease and health areas, but those can be applicable to nutrition as well. So here are some summary thoughts from day 2. And this actually dovetails very well with what Cindy indicated in her conclusion from day 1. We really need to have a systems approach. We really need to think about a systems approach. So I mentioned the word systems probably about 6 times on the slide, because that's to emphasize that nutrition is really affected by this wide range of systems, complex relationships, and they cross many different scales. So you can't just focus on the genetic scale. You can't just focus on the microbiome or the behaviors. You really have to see how these all tie together. Otherwise, you really risk bias, lack of inclusion, diversity, and really understanding how these things interplay. Of course, tech alone is not going to be the solution, but it can help untangle these complex relationships and these complex systems. So the key is to really understand how these complex issues are interlaced, and how technology can help the understanding of these things. There's a need for a lot more diverse data and data sharing. That concept was really echoed multiple times throughout the day yesterday. And we're still in the early days. We're really just scratching the surface. So we got some great examples, but there's still a long ways to go. So there's many things, many different opportunities. The question is, how will this evolve? Will it evolve in the right direction, or will it evolve in the wrong direction? The wrong direction is you're actually going to introduce bias, you're going to worsen disparities, and all that bad stuff. So it's key to have transparency of methods and data. You really have to understand what methods are being used and what data is being used to understand where the insights are coming from. So we want to talk about specifics. So it's important to talk about precision nutrition, the general concepts, but we really need to know how all of these things are actually being implemented. And speaking of implementation, we're now moving on to day 3, where we can talk about some of the specifics in terms of implementation and translation. So I'd like to pass on the virtual microphone to Robin McKinnon of the U.S. Food and Drug Administration, the FDA, for her expert moderation for day 3.